All right, can everyone hear me? We're gonna, oh yeah, that's mine though.
Yeah, okay.
I'll take my coffee. Okay, great. So we're gonna get started with the second session, which is pretty different than the first session. Now we're gonna be shifting gears a little bit and talking about specific phenotypes, particularly psychiatric phenotypes. And I'll be the first of three speakers. And I'm gonna be talking about some studies that we've recently gotten Trey involved in, but sort of a long-going set of studies surrounding rat behavior, especially with respect to addiction. So very broadly, my lab is interested in understanding the relationship between polymorphisms at the DNA level and differences in observable behavior. And largely the approach that we take to that is the standard GWAS approach, which Trey very elegantly described for you in his introductory remarks. And our idea is that as we associate DNA-level polymorphisms with observable differences in behavior in both animals and humans, that we might begin to understand the role of specific genes and their protein products and how those alter the behavior of individual neurons and how those alter the behavior of brain regions and structures as they interact with one another to produce the output of the brain, which is, after all, behavior. And mostly, we take a sort of a single gene approach to that. That is, we use the classical GWAS approach. We identify particular genes for which we have really robust statistical evidence that they're causally related to these behavioral differences. And then we take advantage of the fact that we're working in a model system to manipulate those genes and actually demonstrate causality and begin to get into mechanism. All right? But I have a long-standing interest, actually, in also understanding at the network level how these genes might interact with one another, and kind of a deep belief, which it turns out is shared by a lot of my colleagues doing these sort of GWAS approaches to behavior, a deep belief that really to understand what we're learning from these studies, we have to begin to integrate those genes and understand how they talk to one another, how they maybe have superadditive relationships with one another, et cetera. And so as we sought to renew this center, which I'm going to be describing to you in the next slides, we brought Trey on board because we realized we really needed outside expertise, demonstrated credible expertise in taking the things that we're finding from these GWAS studies and bringing them to the next level where we really could view them both in terms of the single gene approach, but also network approaches. And so, as I said, we have had a center which was originally funded in 2014, long-running center that focuses on analyzing behaviors related to drug abuse, and in particular in using the rat rather than the more classical mouse as a model system. And the motivation, which will be lost on most of the people in this audience, for using rats rather than mice is really that rats demonstrate much more sophisticated behaviors relative to mice. And so particularly as we develop an interest in complex behavioral traits relevant to addiction, like drug self-administration as a primary example, it's important to use a model system where the animals actually will exhibit that behavior consistently. And so in mice, it's a bit of a struggle to get mice to press a lever to get IV cocaine, which is a strong indictment of mice, frankly, because if you can't figure that out, you're really not, you're not thinking very clearly. But rats obviously will acquire that kind of a behavior very rapidly, and rats can understand pretty sophisticated operant schedules where different levers give different consequences, and those consequences might change over days, and et cetera. And so rats are really a preferable model system, but because of the technologies surrounding mouse, people had been hitting their heads against the wall for 10, 20 years trying to do genetic studies in mouse models where they really couldn't get the behavioral paradigms they were interested to work very well in many cases because there was a perception that you couldn't do these things in rats. And so part of the initial mandate of the center was to try to get the kinds of tools necessary to do GWAS at scale in rats rather than mice. And we've been pretty successful there, I think, in the last few years. And so, as I said, as we went in for a renewal, we started to try to bring on new things like this network focus that I'll be describing. It's important to appreciate that to do GWAS in an animal population, you need a genetically diverse population. And so we've taken advantage of the one probably optimal genetically diverse population in rats, and that's this NIH heterogeneous stock. It was created in 1984, so some time ago, by mixing 8 inbred strains, which at the time were selected for their genetic diversity. Pretty accurately, it turns out, despite how little they knew about genetic diversity back then. And they were intermixed over a series of a few generations that are easy to imagine, and then they've been maintained as a large outbred population ever since. So now they're at generation 85 since that original mixing, okay? And that means that these haplotypes, these large blocks, which were unique to each of the 8 bred strains, have had 85 generations to mix, to recombine, breaking them big blocks down into smaller and smaller and smaller pieces. And that means that if we see an association between a marker that discriminates between some of the inbred strains and other inbred strains, that marker must be extremely close to the causal polymorphism to still be associated with it, because if it was far from that, it would have been separated by successive generations of recombination. So that's, of course, the strength of human GWAS, that they use what they call unrelated, which is to say distantly related human beings, which have large numbers of accumulated recombinations that have broken haplotypes down into little blocks. It's actually hard to recover that in an animal system where people have tended to emphasize inbred strains, and where even if they were trying to maintain outbred strains, they've tended to have severe population bottlenecks, which have led to a lot of inbreeding. So this population is really critical to those efforts because it has genetic diversity. And happily, unlike the situation in human genetics where you have a huge preponderance of rare versus common alleles, which are thought to be driving a lot of the sort of unmappable variability, in this population, all of the alleles should be common because their input frequency is about 12.5%. Okay, obviously new mutations are little asterisks for that statement, but nevertheless. So that actually is an advantage to mapping in a population like this relative to a natural population like humans. So the structure of the initial project is relatively simple. We're sending these rats to different phenotyping centers. The phenotyping centers do elegant phenotyping, which I won't particularly describe given the audience, but very, very elegant behavioral phenotyping, sometimes keeping the animals, in the case of one center, for 4 months, where they're essentially phenotyped on a daily basis. So a huge amount of behavioral data are generated. And then they come to us where we're genotyping the animals. We also get brain regions, which we're doing RNA-seq so that we can map expression QTLs, as I tell you in a few slides, and then trying to integrate that, but again, mostly in the initial funding period with the worldview that we were trying to identify individual genes that were associated with these behavioral differences. In addition to the behavioral phenotyping, which is shown up here, we actually have a huge physiological component, which could be of interest to some of the people in this audience. So we said, although we're really interested in drug abuse-related behaviors, once we've genotyped and generated these animals, we might as well collect various tissues and other kinds of data from these animals, because we were able to identify a number of collaborators that were interested in those sorts of things. So in fact, we sort of have a disassembly line for these rats when we're done with the behavioral testing, and we send tissues off to about half a dozen labs, or maybe more like a dozen labs, where various kinds of phenotyping is also done. Trey correctly said that it's inexpensive and efficient to genotype human subjects at millions of polymorphic markers, and that's one of the cornerstones of GWAS. That's not the case for model organisms, particularly for rat. As we embarked on this study, there were no good platforms to do genotyping at scale for lots of SNPs at a cost-effective price point. We've actually turned to sequencing, and I won't describe it in detail, but we use this approach, genotyping by sequencing, which we adapted from agricultural purposes, where we do sequencing at particular loci, maybe like 1 to 3% of the genome selected using restriction enzymes. We sequence that relatively deeply to get genotypes, and then we can impute out using standard imputation pipelines to the whole genome. And that's been one of the things that's really allowed these studies to move forward in rats. So we've started to do the analyses from the first few years of funding. We used body weight and various traits related to body weight as one of the sort of toy phenotypes. So it was a phenotype that we were able to measure in all of the different centers. So at an early time point, this is actually data from about a year ago now, we already had more than 3,000 animals that had been phenotyped for body weight, body length, BMI, fasting glucose, various traits related to that. Actually, we had weighed various fat pads, specific fat pads that we had gotten weights for also. And as you see, we have lots and lots of, so here's the threshold for genome-wide significance. We have lots and lots of genome-wide significant hits for these traits, and they're prominent examples of pleiotropy where various different related traits related to body morphology and weight map to the same locus. So whereas in Trey's example, there was sort of not very many significant things, and it was the idea was to harvest something from the things that didn't cross the threshold for significance. We have the somewhat happier situation that we've got lots of significant things, but we'd nevertheless like to stitch those together into networks. And then you can see there's a huge number of things that are, as Trey described, sort of below the threshold so that we could confidently say any of those results are significant. But there's probably lots and lots of true positives there also, and so we'd equally like to harvest that kind of information. And we're generating data like this for a variety of different traits, and I'm just throwing up these body weight traits as an example. For some of the specific traits, We ultimately are going to have samples anywhere from 1,600 to 3,200 animals. But again, because those data are still being collected, we've done some preliminary analyses. So this is Hao Chen, one of the projects, where he's looking at IV nicotine self-administration. So these animals are pressing levers to get IV nicotine. And as they escalate their administration, we can already see in the first 700 animals, we can already see genome-wide significant hits. And when we look at those in locus zoom, so in other words, zooming in on this peak here, which is a genome-wide peak, you can see that a small number of genes are associated with each of those individual peaks. And so one of our challenges will be to figure out which of the genes in that peak region we think is really the causal gene. And indeed, networks may be helpful in that regard also, because it might be, you can imagine. As I said, we are looking at gene expression in brain regions that we think are relevant. Some of the collaborating grants that I'll describe in a few slides are looking at other gene expression in other tissues as well. So far, we've surveyed 5 brain regions, and in the renewal, we hope to survey 5 additional brain regions. And that allows us to map expression QTLs or EQTLs. So in the same way that you could take any biological trait like body weight or nicotine self-administration and you could map that, we can also measure en masse gene expression traits, and then we can map each of those gene expression traits and identify loci that are associated with heritable variation in the expression of each of those genes in each relevant brain region. Okay, so that's another thing that we're doing. And of course, you can imagine that if you have a map like this and you're wondering which of these genes might be causal, knowing that one of them had heritable gene expression differences might give you a leg up in identifying the causal gene. Indeed, we're also starting to look at single cell, and so Francesca Talise, who's in the audience somewhere, has a pilot grant from our center, and we have a separate R01 that we're hoping will be funded, where we're going to start looking at RNA-seq of a single-cell RNA-seq and then also single-cell ATAC-seq so that we can get a more nuanced understanding of exactly where these heritable gene expression differences have their consequences. So to talk briefly about phenotypes, which I've largely skirted, but in fact is the entire thing motivating this center, we're interested in phenotypes like delay discounting as an example. So delay discounting, you might have heard about the marshmallow test in some kind of a freshman psychology class you took where you tell kids you could have 1 marshmallow now or 2 marshmallows in 5 minutes, and the internet is full of wonderful videos of kids freaking out as they try to make that very difficult decision. And you can do exactly the same thing with animals, it turns out. So you can teach them that one lever will get them an immediate reward. These are some modestly water-deprived animals. They'd like to get water. One lever will get them some water right now, but the other lever, with some predictable delay, will get them a larger amount of water. And you can then titrate either the delay or the disparity between those 2 rewards, and you can basically establish how an animal devalues a reward depending on how long they have to wait for it. Okay, so all of us do that. A reward you have to wait for should be worth a little bit less than a reward that you can get now. But we have, it turns out, different curves for how we devalue that. And so some individuals, both animals and humans, are less impulsive. That is, they value future rewards and they assign them a value that's relatively high despite the delay, and others devalue them more steeply. Okay, that is, they choose that 1 marshmallow because waiting for 5 minutes is too much. And those are heritable traits, and in fact, we've looked at some of the inbred strains that were used as the foundation for this population and characterized delay discounting in each of those inbred strains and found that the heritability of delay discounting was somewhere on the order of about 60%, 50 to 60%. So it's really very strongly heritable in animals. Obviously, in humans, those questions are a lot more complicated because they're typically put in terms of money, not water. And money has kind of different values to people. And in particular, you might be broke or you might already have money, in which case getting $5 now or $10 later has a very different interpretation. We also are getting nice hits for delay discounting. So here's a genome-wide significant hit again, just kind of emphasizing that we are pulling out genes, even with the relatively early analyses, we're pulling out genes or regions that contain a few genes that are strongly associated with these traits. And so we really have something that we'd like to harvest here. In fact, we're also interested in delay discounting in humans, and one of the themes of the project that Trey and I hope to incorporate into the renewal is that we'd like to use networks as a way of stitching together what we're finding in animals and what we're finding in humans. And so we have parallel studies going on looking at delay discounting in humans, and we've actually collaborated with 23andMe, who's allowed us to ask these same delay discounting questions of large numbers of human subjects. So in our initial kind of pilot study. We did 25,000 subjects. We've now got funding and an agreement with them so that we're going to increase that to more than 150,000 subjects. And so that allows us to do genome-wide association studies for what we believe is the same trait in a human population. And then, of course, you might imagine that you could find the same gene in both the humans and the animals. That would be the simple story that big journals like. But more likely, we're going to find different genes in the animals and the humans, not because animals and humans are really that different in terms of delay discounting, but because the existing variation in animals and humans and the poor power that we have in both systems to identify all of the causal factors will lead us to identify probably somewhat or even non-overlapping sets of genes. And so here, just a picture of some of the people involved in the 23andMe study, that initial study of 23,000, already identifying one genome-wide significant hit. And if you take Trey's idea that some of these sub-threshold hits may be interesting, probably additional hits also. And so as we increase the sample size, we hope to get some convergence. Again, might be at the level of individual genes, but if we could superimpose these results onto a network, we might identify parts of the network that were important for both of these, even if it wasn't the same individual genes. The success of a bureaucracy, for better or worse, is kind of measured on whether or not that bureaucracy grows over time, okay? So it's a sort of a very cynical parable that I was told once. And so in the same way, you might measure the success of a grant or a center grant based on whether or not it produces additional grants, okay? And I don't fully endorse that view of the world, but it's one way you might look at it. And in fact, over the first 4 years, we will be able to foster lots of additional grants developing this idea of using the rat as a platform for genetic studies to study both behavioral and other kinds of traits. And so here's a list, and it's obviously too small to see, but in particular, there's 6 U01 grants that are sort of spin-offs of this that seek to use rats as a behavioral model. And then there are also a number of R01 and other kinds of grants that are taking advantage of those tissues, which I briefly mentioned that we're producing. So there's sort of a big group of people starting to work in the rats, starting to use this common population and these common tools of GWAS to generate really impressive data sets. And I think that makes it fertile ground to try to harvest something at the level of networks as well as individual genes. Just to kind of further plug the interesting things we're doing, we're also looking at cocaine self-administration. And in the case of cocaine, it seems that we're really identifying 2 different populations, one that's really susceptible to escalating their intake of cocaine over an extended period of access. So this is 14 days where they have 12 hours of access to IV cocaine, and you see that one group really increases dramatically the amount of cocaine they're taking, and one doesn't. So these are the kind of really interesting and I think arguably interpretable phenotypes that we can look at in rats. The physiological collaborators, I see Iris standing up here, so I think I'll wrap up. And we're also, more physiological stuff that's wonderful. We've been very careful to store the data from the outset in a very well-organized database, and I think that might be important for some of the studies that I hope to engage people in this audience with. All of the animals have a unique ID, so a microchip that they're given just at weaning, and they're followed based on that microchip ID so that all of the information we have about them, that's pedigree information, that's behavioral information, that's physiological, genotype, gene expression, microbiome, et cetera, information is all in a database. We can track it to the animal. And we also store the raw files for all of these animals. So as we collect behavioral data, you can imagine that's a very involved protocol. But what we're doing is storing the raw files that we get from these data acquisition behavioral apparati in the database. And then we have scripts that extract the phenotypes that we're interested in, but we can then rewrite or retweak those scripts to extract different phenotypes as our questions about the behavior evolve. And so it's, in that sense, a powerful database. We're also making a lot of this data available for people that want to do sort of straight-ahead GWAS approaches on Gene Network, which is a publicly available database that allows people to look at the data, do mapping kinds of things, do correlated trait mapping, can pull out principal components from multiple traits and map those, et cetera. All of this on this Gene Network website. So the data are available in a few different forms. And then finally, just to reiterate the thing that I think I've been plugging throughout this talk, is that we're working with Trey to reanalyze the GWAS results, sort of doing that based on the nearest SNP as a way of identifying gene, but also using an approach called TWAS that I haven't had time to describe, but transcriptome-wide association analysis, where we could impute the presumed transcript levels in various tissues of interest and use those rather than the SNPs as the substrate for the association mapping. And then also trying to integrate with human data sets that are relevant, and then some even fancier stuff. So I think really the purpose of my being here and talking to this audience is to say we have this resource, a really potentially very powerful resource because it's an experimental system, a resource where we can do directed experiments or get tissue much more easily than you could do in humans. And we're very enthusiastic at moving forward with collaborations to enhance the impact on psychiatric genetics. Okay, so I'm going to stop there. We could do maybe some questions, Iris? I don't know how long I talked. Yeah, maybe some questions, right?
Okay, I have a question for you, Abe.
Okay.
So actually, how did you pick what are the sites that how do you select the sites for targeting?
Oh, so you mean for the genotyping?
Correct.
Yeah, so we're not concerned. So we're just using some restriction enzymes, which will randomly subset the genome, and that way we can get deeper coverage at fewer sites. And we're able to do that because there are haplotypes that exist in any population, including this population. And so the haplotypes mean that we don't need to get all of the genotypes in a block. We just need to get enough genotypes that we can impute which is the founder. Yeah. It's a trick. Like for the purposes of anybody understanding what we're doing, we could have used a chip, but we didn't.